Jai Hind, dear children. Welcome back to yet another session of biology online class. In the last two videos, we have discussed about the autotrophic nutrition in detail. You have learned how the inorganic substances like water, carbon dioxide, sunlight, etc. are obtained by the plants from their surroundings and the process of photosynthesis. And you have seen the experiments which shows that carbon dioxide, sunlight and chlorophyll are very much necessary for the plants for the process of photosynthesis. And I hope all these topics are clear for you now. Now, today in this video, I am going to show you how to prepare a temporary slide of leaf peel to observe the structure of stomata under the microscope. You have to do this experiment as lab activity. I hope all of you are having your lab manual, no? So all of you please take your lab manual with you and take experiment number 8. What is the experiment? To prepare a temporary mound of leaf peel to show the stomata. For doing this experiment, some materials are required. It is mentioned in your lab manual. So what are the materials required? Plain slides, watch glasses, forceps, needle, blade, brush, water dropper, dilute saffron in solution and glycerin, and the compound microscope to observe this slide. Here we have taken some leaves which was already cleaned with water. Here you can see 3 watch glasses. In the first one we have taken plain water, and in the second one dilute saffron in solution, and in the third watch glass again plain water. Here you can see the upper surface of the leaf, which is covered with the upper epidermis, and the lower surface of the leaf, which is covered by the lower epidermis. You have already studied about the structure of the leaf, in which there is an upper epidermis and a lower epidermis. And you have studied that a large number of stomata are present on the lower surface of the leaf, more than that on the upper surface. First of all, what we have to do is to peel off a small part of lower epidermis in which stomata are present. So for this, we have to tear the leaf from the leaf margin and fold the teared portion and pull it in the opposite direction of the veins of the leaves. Do it carefully in such a way to separate the epidermal layer from the leaf. Now you can see a thin transparent layer, and this is the lower epidermis in which large number of stomata are present. So we can use this thin epidermal layer to observe the stomata under the microscope. Now we have to cut and separate the epidermal peel by using a blade. Now with the help of a brush, transfer this peel into the watch glass containing dilute saffron in solution for 30 seconds or 1 minute to stain the peel, that is to give color to the cells. After one or 2 minutes, with the help of a brush, transfer this peel from the saffron solution to the watch glass containing plain water again to remove the excess stain in the peel. After this, transfer the epidermal peel on a clean, dry glass slide. Now, add 1 or 2 drops of glycerin with the help of a dropper on the epidermal peel and cover it with a clean cover slip. Now hold the cover slip on one side and place it vertically on the slide by holding one end with the help of a needle and cover it over the peel to avoid the formation of air bubbles. Now the slide is ready to observe under the microscope. For this, we are using a compound microscope. You have studied in the 9th standard the structure and functioning of a compound microscope. Here you can observe the epidermal peel using a 10x objective lens. Here you can clearly see numerous small pores scattered in between the epidermal cells, right? And these small pores are called as stomatal pores or stoma. Now we can observe the same slide by using 45x objective lens, by which we can see some more magnified. What you can see now? Here you can clearly see the stomatal pores, which are guarded by two kidney-shaped guard cells, and these guard cells control the opening and closing of stomata. And these guard cells are surrounded by some irregular subsidiary cells, and around these subsidiary cells you can see the epidermal cells. You can see some stomata are opened and some stomata are closed. This is how you can prepare the temporary slide of a leaf peel to observe the stomata under the microscope. So when you are back to school, you can do it in the lab. Meanwhile, you have to write this experiment in your lab manual and draw the pictures. So you have to write the aim, materials required, the procedure, observation and inference in your lab manual, and draw the diagrams in the left unruled page. So that is all about the autotrophic nutrition. Next, we can discuss about the heterotrophic nutrition in detail. Heteros means others and trophic means nourishment. We have already discussed about the different types of heterotrophic nutrition. Mainly there are three types of heterotrophic nutrition. They are saprophytic nutrition, parasitic nutrition and holozoic nutrition. This classification is based on the type of food, availability of food and the mode of obtaining the food by different organisms. Different organisms obtain different types of food in different ways. The first one is saprophytic mode of nutrition. The term sapros means dead. So some organisms obtain nutrients from the dead or decomposed organic matter. For example, fungi, bacteria, etc. Next one is parasitic mode of nutrition. Parasitic means living on another organisms. Some organisms live in the body of other organisms and obtain nutrients from that host organism. For example, you would have heard the people having worms inside the stomach, no? So here the worms are the organisms which are living in our body and obtain nutrients. Like this, lice is another organism which belongs to insects, which is present on our hair and obtain nutrients from our body. The third one is holozoic mode of nutrition. What is holozoic nutrition? The term holo means as a whole. So holozoic means some organisms intake complex organic substances as food and break down this complex food substances into simpler food molecules with the help of some enzymes. And then it is utilized by the body. If we take human beings as the example, what do we eat? We eat things like rice, wheat, meat, etc. as food, right? All these are complex food substances. But we need simpler food substances like glucose, proteins, etc. But we do not usually intake glucose. Instead, what we eat? We eat complex food substances like rice, wheat, meat, etc. But the cells in our body cannot utilize this rice, which is a complex organic substances. So the rice needs to be broken down into simpler food substances, that is glucose. Then only our body can utilize it. Like this, all other nutrients necessary for our body like proteins, fats, etc. are obtained by the body from this complex organic food substances. So basically, the holozoic nutrition means the animals intake complex food substances and break down into simpler food substances and then utilize it. The best example for holozoic nutrition is human beings. Most of the animals which we see around us are holozoic. All animals depend on plants or other animals for their food. Some animals intake food from the plants directly, and some animals intake the flesh of those plant-eating animals. So we can say that heterotrophs directly or indirectly depend on plants or autotrophs for their food. Accordingly, there are different types of heterotrophic organisms that are herbivores, carnivores, omnivores, etc. Let us now discuss about the holozoic nutrition in detail. That is, how the food is intaken by certain organisms and how it is utilized by the body. There are different ways by which different organisms obtain food from their surroundings and break down into simpler food molecules and utilize it. In case of unicellular organisms, that is single-celled organisms, the food may be ingested from any part of the cell surface and they perform intracellular digestion. That is, the breakdown of complex organic substances into simpler food molecules takes place inside the single cell itself. That is intracellular digestion. But in case of multicellular organisms, different part of the body becomes specialized to perform a particular function. Holozoic nutrition involves 5 steps in order to convert the complex food substances into simpler form. They are ingestion, digestion, absorption, assimilation, and egestion. The first step is ingestion. Ingestion means intake of complex food substances. Different organisms intake the complex food in different ways. Next step is digestion. Digestion means the conversion of complex food into simpler form with the help of certain enzymes. The next step is absorption. Once the food is converted into simpler forms, that can be absorbed into the cells through blood. Assimilation is the next step. Assimilation means when the food is absorbed by the body cells, it is utilized for obtaining energy needed for various activities of our body. The last step is egestion. Some of the complex food that we eat remain undigested and that has to be removed from our body. This process is called egestion. So all these processes, that is ingestion, digestion, absorption, assimilation and egestion, constitute the holozoic mode of nutrition. Now we can discuss about the holozoic nutrition in simpler form of organisms, that is unicellular organisms. We can take the example as amoeba. You are all familiar with how amoeba looks like, right? As you have already studied in the lower classes. Here you can see the picture of amoeba. Amoeba is a microscopic unicellular organism and it belongs to the group Protozoa. And it has no definite shape. It can change its shape, but it is having a definite nucleus and the cytoplasm consists of all the cell organelles. Amoeba perform holozoic mode of nutrition and it intake other unicellular organisms, example paramecium, etc. as its food and obtain nutrients from them. Now let us see how different steps of holozoic nutrition takes place in amoeba. The first step is ingestion. Ingestion means intake of food. When any food particles or any other unicellular organisms like paramecium reach near the amoeba, It stretches its cell membrane and produces two finger-like projections called pseudopodia. What is pseudopodia? Pseudo means false and podia means food. That is false food or temporary food. So this pseudopodia is the temporary extension of cytoplasm. So whenever any food material is present in its surroundings, it stretches its cytoplasm and produces pseudopodia and capture that food. And this process of intake of food by amoeba with the help of pseudopodia is known as phagocytosis. And when the ends of these both pseudopodia touches each other, the cell membrane at that point dissolves and forms a food vacuole in which the food is encaptured. Now, the second step is digestion. So, as we have discussed that digestion is the breakdown of complex food substances into simpler food molecules. Now, how digestion takes place in amoeba? The cell organelles called lysosomes present inside the amoeba secrete certain enzymes into the food vacuole. So, what are lysosomes? You have already studied in 9th standard that lysosomes are also called as the suicidal bags as it contains powerful enzymes. So, when the lysosomes release these enzymes into the food vacuole, the complex food gets break down into simpler forms and thus the food gets digested. Now the food gets digested and the next step is absorption of these nutrients. From the food vacuole, the nutrients obtained as a result of digestion gets into the cytoplasm by the process of diffusion. What is diffusion? The transfer of molecules from a higher concentration to a lower concentration. So the nutrients are there in the food vacuole in higher concentration and from the food vacuole, these nutrients will diffuse into the cytoplasm where it is in lower concentration. Next is assimilation. In this process, the digested and absorbed food molecules are converted into various constituents and are stored as food reserves in the cell and are utilized by the cell for various cellular activities. So that is assimilation. The next is egestion. After the digestion process, some undigested food remainings are there in the food vacuole and this has to be thrown out. For that, the undigested food containing food vacuole moves towards the surface of the cell and fuse with the cell membrane and throws out the undigested food by bursting the food vacuole. This process called as egestion. These are the 5 steps by which nutrition in amoeba takes place. Another unicellular organism is paramecium. You are familiar with the paramecium as you have already studied about the paramecium in the lower classes. This paramecium also performs holozoic mode of nutrition. It feeds on other microorganisms. Unlike amoeba, the paramecium has a definite shape and the body is surrounded by hair-like structures called cilia. This cilia helps in the locomotion of the paramecium and also this movement of this cilia brings the food particles towards the oral groove or otherwise called as mouth-like structure through which the food is intaken into the paramecium. After entering into the oral groove, there forms the food vacuole. Here also, the lysosomes secrete the enzymes into the food vacuole and this food gets digested inside the food vacuole. The digested food is absorbed into the cytoplasm and is utilized by the cell for various cellular activities. And the undigested food is thrown out from the body through another opening called as anus. So children, I hope you all got an idea about the holozoic nutrition in unicellular organisms, right? So I hope all of you understood the contents that we have discussed today. All of you write the experiments in your lab manual and draw the diagrams and read the textbook thoroughly and prepare the notes. Thank you.